Hey guys, Miss Cote here, and this is video number 2 of the cell unit. In this episode, we're going to start looking at each of the organelles in a eukaryotic cell. You're expected to know the structure and function of each one of these organelles, along with how they can work with other organelles to get their jobs done. I'm just going to let you know that College Board likes to structure their questions about organelles by talking about symptoms of a disease or disorder, and you need to identify the organelle that's not doing its job and therefore causing the disease or disorder. So let's get started. First, let's start with the nucleus and the ribosomes. These two organelles work together to produce proteins for the cell. As I said in the last video, in the eukaryotic cell, the nucleus is where most of the DNA is contained. The DNA holds all of the instructions that make you, you. But in order to make the proteins that these instructions code for, a copy of the instructions need to be sent out to the ribosomes, which is where the proteins are put together. When looking at a cell, the nucleus is the most prominent organelle. You can usually see a nucleus when looking at a cell under a microscope. Here we have the nucleus in purple, surrounded by a double membrane called the nuclear envelope, and this separates the inside of the nucleus from the cytoplasm. Remember in the last video, I talked about the importance of creating different compartments in the cell. Embedded in the nuclear envelope are nuclear pores, complexes that regulate what goes in and out of the nucleus. If you look closely at a pore, it spans across both membranes. Inside the nucleus, the DNA, along with some proteins, are arranged into units called chromosomes. When the cell is not dividing, we refer to the chromosome as chromatin, which is actually more of a relaxed, stringy structure. When the cell is preparing to divide, the chromatin condenses and coils up to form what you know as a chromosome. Also inside the nucleus is a darker structure called the nucleolus. The nucleolus is responsible for making ribosomal RNA. You may remember it being called rRNA. The ribosomal RNA will exit the nucleus and go out into the cytoplasm, which makes a nice segue into the next slide, which brings us to the ribosomes. The ribosomal RNA that leaves the nucleus teams up with proteins to make the ribosomes. Ribosomes are the organelles that make the proteins. If you have a cell whose job is to make a lot of proteins, then you should expect to find a large number of ribosomes inside it, as opposed to a cell that doesn't make a lot of proteins. You may remember that ribosomes can be in one of two places. They can be free, floating around the cytosol, or they can be bound to the endoplasmic reticulum. Depending on where they are, free or bound, the proteins they make are destined to different places. Free ribosomes make proteins that are used inside the cell, while proteins made on bound ribosomes will be used in the cell membrane or secreted outside the cell. Also, the ribosomes can switch between being free and bound. College Board likes to ask questions about the differences between free and bound ribosomes. The structure of a ribosome is quite simple. It's made up of two subunits, a large and a small subunit. These structures are usually separated and come together when they need to make a protein. The whole process of protein synthesis from DNA to ribosomes will be discussed in more detail in Chapter 17. This brings us to the next set of organelles that's referred to as the endomembrane system. These are the membrane components of the cell, starting with the nuclear envelope, ending with the plasma membrane, and including all of these organelles in between: the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles. What's important to remember is that all of these organelles are made up of membranes, phospholipid bilayers, and these membranes are basically interchangeable, meaning you can take a piece of membrane off of one organelle and attach it to another organelle. They may not be completely identical, but they can be modified to fit in with whatever organelle they're currently attached to. When these membranes break off of an organelle and travel to another organelle, these membranes form little sacs or bags called vesicles. And as we will see, the vesicles can carry materials from one place to another. The endoplasmic reticulum, or ER for short, is continuous with the nuclear envelope. So the ER is located right next to the nucleus. There are two regions of the ER, smooth and rough ER. The main difference between the two structurally is that smooth ER does not have ribosomes attached to the surface, where the rough ER does. And that will give each type of ER different functions in the cell. So let's look at a picture of the ER. Here we have the ER colored in blue. If you look closely, it's basically a continuation of the outer membrane of the nuclear envelope. The little brown dots represent the ribosomes, and they're found on the outside of the rough ER, but not the smooth. The ER forms a compartment separate from the cytoplasm. The inside of the ER is referred to as the lumen, and this creates a space so the ER can do what it needs to do. The functions of the ER vary depending on if it's smooth or rough. The smooth ER is responsible for a number of things, including making lipids, metabolizing carbohydrates, detoxifying drugs or poisons, and it also stores calcium ions. So in cells that are responsible for doing these things, you would find an abundance of smooth ER. Because the rough ER has the ribosomes, it's basically a protein factory for the cell. Its main function is to collect the proteins that are being made from the ribosomes inside the lumen. From there, they're packaged into transport vesicles and shipped off to either the cell membrane or to the Golgi, where they can be further modified. So any cell whose job it is to make proteins to be secreted will have a lot of rough ER. Which brings us to the Golgi apparatus. The Golgi is made of flattened membranes to form sort of a shipping and receiving center. The transport vesicles that come from the ER bring the proteins into the Golgi where they can be modified. The Golgi can also make some polysaccharides as well. All of these products can be stored here until they're ready to ship out to their next destination. Here is our Golgi apparatus shown in, let's call this teal. Here we have a transport vesicle bringing a protein from the ER. The vesicle will fuse to the membrane of the Golgi and become part of it. And when the product needs to be shipped out, it will bud off from the Golgi and become a vesicle. Notice that there is a directionality to the Golgi. The receiving end is called the cis face, while the shipping end is called the trans face. So like the rough ER, any cell that needs to make proteins for secretion will have a good amount of Golgi membranes. Some of those vesicles that pinch off from the Golgi contain hydrolytic or digestive enzymes and become lysosomes. You were probably taught that only animal cells have lysosomes, but new research suggests that plant cells may have them as well. Inside the lysosome is a more acidic environment where these enzymes like to work. This is actually a good thing because if a lysosome accidentally breaks open and the enzymes leak out, they won't necessarily destroy the cell because the enzymes will not be very active. Remember that enzymes like to work at a particular pH. However, if many lysosomes all break open, that could definitely destroy the cell. The job of a lysosome is to use those enzymes to digest things. One example would be over here on the left, where food molecules are being taken into the cell through a process called phagocytosis. A lysosome can fuse with a food vacuole, and the enzymes can digest and break down the food into smaller molecules, and those can be reused. This is frequently seen in one-celled organisms that use this method to get their nutrients. We have white blood cells called macrophages that use this method to gobble up and break down any bacteria that have gotten into our bodies and are trying to make us sick. On the right, lysosomes can be used to digest any organelles that may be damaged. We can break them down into smaller parts and then recycle those parts. This is a process called autophagy, or self-eating. Lysosomes are very important because they keep the cell working cleanly and efficiently. If the lysosomes are not working properly, then there will be an accumulation of stuff, which can lead to problems for the organism. Last are the vacuoles. Again, you may have learned that only plant cells have vacuoles, that large storage structure that takes up most of the plant cell. but animal cells can contain vacuoles as well. Vacuoles are large vesicles that come from the Golgi root, and they can have a variety of functions depending on what type of cell they're in. We just saw an example of a food vacuole when we were talking about lysosomes. Some freshwater unicellular organelles have specialized vacuoles called contractile vacuoles that are used to pump out fresh water that enters the organism through osmosis. And we'll talk about that in the next chapter. But you should all be able to recognize the large central vacuole found in plant cells. This type of vacuole is mainly used for storage for water, salts, and other organic compounds. Again, when we get to the cell transport chapter, we'll talk about how the amount of water being stored in the central vacuole can exert pressure on the cell wall. And that covers all of the organelles grouped into the endomembrane system. It's very important that you understand how these organelles work together to create a very complex and dynamic system in the cell. There's constant movement of the membrane, flowing from one organelle to the next, moving proteins and other molecules around the cell. And that's where I'm going to end for today. We'll continue our tour of the organelles in the next episode, where we'll discuss the organelles involved with energy and structure. Any questions you may have about this episode can be brought up at the beginning of class. So until next time, be a good human.